Okay, so continuing on our discussion of organelles within what kind of cell? Not prokaryotic, but eukaryotic cells, right? Because if it doesn't have organelles, it's prokaryotic. If it does have organelles, it's eukaryotic. This is the mitochondria. A lot of folks know this is the powerhouse of the cell, and for right this minute, that's good enough. Basically, what you should know right now is that mitochondria use sugars to generate ATP. ATP is cellular energy. That's what mitochondria do. Okay, they make ATP. That is their job. You should maybe also know that mitochondria are roughly the same size as a bacterial cell. So generally, like I was saying earlier, eukaryotic cells are much larger than bacterial cells. In fact, they have many of these, roughly bacteria-sized mitochondria within them. I'm not as worried about the terminology of the specific anatomy of the mitochondria right now. Don't worry, we'll get to that probably next week. Here is a chloroplast. Okay, and these only exist within plant cells. That was one of the questions on one of your labs. Chloroplasts are only within plant cells. And what chloroplasts are doing is they're taking energy from the sun and using that energy to make sugar. Why are they making sugar? Well, they're making sugar so that their mitochondria can make ATP from it, which again, we're going to talk about next week. This is a chloroplast. It is another kind of organelle. It only exists within plant cells. Plants and some protists, but neither here nor there. Okay, you should know the function of this organelle, though. Taking light from the sun, making sugar. This is the extracellular matrix. Okay, so here you see the plasma membrane that we talked about in the last lecture, and you have fibronectin and collagen fibers and all of these things that are helping to anchor the cell. It's helping the cell to communicate with other cells. This is sort of the area outside of the cell. Don't worry about like proteoglycan. Just know that extracellular matrix is stuff that the cell secretes to help anchor it, to help it communicate, to help the cell do different things. And obviously, the extracellular matrix exists in both prokaryotes and eukaryotes. So we're talking about extracellular matrix. Certainly, your cells are communicating with one another, right? It's the reason that your brain can send an electrical signal and make your hand do this, or make you speak, or whatever. But cells are connected to each other in particular ways. There are 4 kinds of cell connections that we're going to talk about. And we have plasmodesmata here, which is this gap right here. And this is literally, when you have 2 plant cells next to each other, very often they are continuous with one another. Connections, they go through the cell wall, this outer structure here. They go through the cell membrane, or plasma membrane, okay? And they connect 2 plant cells together. Animal cells, we have these things called tight junctions, and you can think of, when you think of tight junction, think of watertight junction. Okay, water cannot escape these. Now think about where you might want something where water cannot escape. Something like, I don't know, perhaps your urinary bladder. You don't want a bunch of urine in the inside of your body. Put cells with a bunch of tight junctions around them. Okay. Excuse me. We also have desmosomes, and desmosomes are going to be like 2 cells sort of being like welded together so that they can form sheets that are sort of they have the ability to be somewhat elastic. Okay, so you're going to see desmosomes on your like skin, okay, or in your muscles. So it's going to form these sheets so that you're still able to move around, but those cells are going to be stuck together. We also have things called gap junctions, okay, and they're kind of like plasmodesmata, but they're different. If you study neuroscience, you will learn a lot about gap junctions. They allow cells to be connected to one another and actually communicate with one another. But yeah, don't worry about terms like I see it says connects in here. Don't worry about that. I just want you to know there are 4 basic kinds of cell-to-cell connections. Plasmodesmata are only in plant cells. You should know about tight junctions. You should know desmosomes, and you should know gap junctions. Not too much beyond that. Just kind of know what they are and maybe know a sentence or two about their function. Where'd my mouse go? Okay, so this is the fluid mosaic model. And in the fluid mosaic model, you have all of this sort of stuff in your plasma membrane, right? It's all there. In the fluid mosaic model, these things are able to move around quite a bit. They're actually able to move from place to place. And if you imagine the phospholipid bilayer as kind of like this movable sort of sea of phosphates and lipids, where these things are sort of not bobbing around, but they're able to move. Okay, fluid mosaic. These things are able to move around inside of this lipid bilayer. Okay, it's not static. Those glycoproteins, those cholesterol, those integral proteins, those are all able to move around the lipid bilayer. Okay. That's the fluid mosaic model. So one of the reasons why these semi-permeable membranes is so important is because of the idea of diffusion. Diffusion is a really big idea. It's something that you should definitely understand. Okay, so if you don't quite get diffusion after this, make sure you let me know so I can explain it in a different way or figure out a different way of saying it, or so that we can at least figure out what's going on. Okay, so it says here, diffusion through a permeable membrane follows the concentration gradient of a substance, moving the substance from an area of high concentration to one of low concentration, blah, blah, blah. Basically what this is saying is that if you put a whole bunch of stuff in one area, it's going to naturally tend to move away from that. So this works better in a classroom, but the way I like to say it is, if I were to tell all of you guys, imagine we're sitting in class one day, and I say, hey everybody, I want you all to go stand in a corner. And this example works way better before COVID too. But imagine I say, everyone go jam yourselves, go get packed back into the corner, and then I left the room for a couple of hours. Do you think that you would stay just sort of jammed in the corner like that with everybody right next to you? Probably not. I imagine you would probably diffuse throughout the classroom until you're roughly equally spaced out. So you would go from an area of high concentration, where I pushed everyone into the corner, to low concentration. The rest of the room, you would spread out, right? And so that's the idea behind diffusion. And you can see whatever this particle is, where'd my mouse go? Whatever this particle is out here is diffusing across this lipid bilayer. Okay, it's high concentration out here, it's low concentration in here, and so it's slowly diffusing across. But the thing is, most things, I shouldn't say most things, but a lot of things cannot cross through that lipid bilayer, which is where the idea of osmosis comes in. So diffusion is solute particles, and solutes are, it's something that's a word you should know. Solute is the stuff that gets dissolved. And so if I were going to make myself a glass of sweet tea, I would take some tea, right? And I would dissolve some sugar in it. So in that example, what's the solute? Solute is the sugar. The solvent in that case would be the tea, or the water in the tea, however you want to think about it. But solute is something that is dissolved in water. So here we have this thing called a semi-permeable membrane, which, by the way, the lipid bilayer of all of your cells is a semi-permeable membrane, meaning that some things can get through it and some things cannot. So this semi-permeable membrane is here. We have some solute particles, whatever these happen to be. And we have more solute particles on this side. And so if we're just looking at the left side here, if diffusion was able to take place, all of these particles would move over from this side to this side, right? because they're in higher concentration here and lower concentration here. And so, hey, there's a bunch of students jammed into the corner here. They're going to move throughout the room. But the semi-permeable membrane prevents them from moving from this side to this side. And so what happens is the process of osmosis. And that is water following the solute. Wherever solute goes, water is sure to follow. And so you can see here, water has followed this solute, and now the water level on this side is much higher. But what's actually happening here is roughly now the concentrations of these two sides are equal, because there's way more water to solute. There's way more water on this side, even though there's more solute. This has huge medical applications, and so it's the reason why you get salt water in your IV drip. It's the reason that there's saline in those IVs and not just pure water. So actually, if you shoot me an email that says, why don't we call it tonicity, Tonicity, tonicity extra credit, and you tell me what would happen if they put pure water into your IV drip. What would happen to your blood cells? Would they become hypertonic? Would they become isotonic? Or would they become hypotonic and explode? I should also explain, these are relative terms. So hypertonic is one thing relative to another thing. And so, you know, it doesn't make any sense to talk about hypertonic in a vacuum. Like if I said, hey, where is my pen? It wouldn't make any sense for you to say like above. Like what above what? What do you mean? You could say my pen is above like my computer. You could say my pen is above the floor, but you can't just say, oh, well, that pen is above. Like above what? Below. That pen is below. Below what? Okay. So whenever you're using the term hypertonic, it is your blood is hypertonic to the solution around it. Your blood is isotonic to the solution around it. Or your blood is hypotonic to the solution around it. So I'm going to go ahead. This video is getting kind of long, but I'll explain these really fast. So hypotonic, okay, that means that there is more stuff inside of the cell. There's more solute inside of the cell than there is in the solution around it. There's more solute in there. Where is water going to go? Remember a few seconds ago I said, wherever solute goes, water is sure to follow, right? And so in this case, water is going into those blood cells. It's going into those blood cells, and they're going to swell up and swell up and swell up, and they're ultimately going to pop, which is what happened to this poor blood cell here. Hypertonic, on the other hand, means that there is more solute outside of the cell. Okay, or you would say the solution is hypertonic to that cell. And so water is going to follow the solute. It's going to leave these cells, and water and the cells are going to shrivel up. Isotonic is the concentration is roughly equal inside and out. And so maybe some water comes in, maybe some water goes out. But isotonic means that concentration of solutes is roughly the same. Okay, and here's a picture with some plant cells, and hopefully you guys were able to see what happens when you put a salt solution near plant cells in lab. We're not going to get into this right now. I'll save that for the next lecture. So yeah, stuff we learned today. Diffusion is a huge one. You should know about those organelles. You should also know about osmosis. That's another huge one. You should know what the term solute means. You should know the term solvent. You should know what the term solvent means. And you should know that wherever solute goes, water is sure to follow. Okay. Yeah, that's long enough.